Sure. YTB323 is a novel autologous CD19-directed CAR T-cell that uses this T-charge platform for production. So as I think most people are aware that CAR T-cells have dramatically changed the way we treat patients with relapsed and refractory large cell lymphoma, and there are a number of publications here at ASH this year about this. But one of the obstacles is that it can take a significant amount of time to produce the CAR T cells. And so novel ways of trying to improve the time from collection to infusion of the CAR T cells is important. So YTB3 is manufactured through an innovative process called T-charge, which not only speeds up the time to delivery, but preserves the naive T memory stem cell phenotype in the final product consistent with the leukapheresis product, and may produce a more longer lasting, more efficacious CAR T cell. And so this was a study looking at, it was a first in human dose finding trial in patients with lymphoma, large cell lymphoma, CLL, ALL, and this presentation really focuses on the diffuse large B cell component. So eligible patients were those patients who have had, this would be third line diffuse large cell lymphoma. They underwent the traditional apheresis, and then they could have optional bridging chemotherapy followed by lymphodepletion and reinfusion of the YTB product. And we explored a couple different dose levels. We looked at 2.5 times to 10 to the 6 cells, as well as 12.5 times 10 to the 6 CAR T cells. And the dose finding is ongoing at this time. So there were about 20 patients that were entered onto this trial, 2 different dose levels, 16 at dose level 2 and 4 at dose level 1. And there was, the results were actually quite good. So we saw an overall response rate of about 80% at dose level 2. 73% of patients achieved a complete remission. 73% of patients remain in complete remission at 3 months post-infusion of the CAR cell. So that was also quite exciting, suggesting high efficacy. So the other important finding was that this product was also associated with a good safety profile. So there was very little grade 3 or higher CRS, very little grade 3 or higher neurological toxicity. Most of the adverse events were in the grade 1 to 2. And so I think this is an important finding. And I think the other important finding here was that the time to some of these events was pushed back significantly. So for instance, the median time to both CRS as well as neurological toxicity was greater than a week, which I think really opens up this therapy to outpatient. The other important finding of this trial was, despite using a 25-fold lower dose of YTB, we get nearly the same expansion that was seen in the JULIET trial with Tisagenlecleucel. So I think these are really exciting findings. This therapy hopefully will continue to be developed and maybe a major improvement in the way we treat patients with diffuse large B-cell lymphoma.